Hey, what's up everybody, and welcome back to the Sanctuary. I'm Professor C, and we're going to talk more A&P today. Specifically, we're going to talk about the central dogma. We're talking about some DNA today. Part 1 of 3 parts. Replication here, transcription next, and translation will be the third. So, let's dive in to some DNA replication. Let's go. Alright, so we need to talk about some genetics here, and I don't want to do it in one big talk or it will just go on and on and on. So I'm going to break it into 3 parts. This will be the first of 3. And as usual, if we want to learn about something complicated, getting the terminology down is critical. So let's take a little trip to the library, as this slide suggests, and let's deal with some terms that we're going to see come up repeatedly in these talks. So the concept of genetics. Now you can read that on your own, I'm sure. The science of heredity, including the study of genes, how info is carried, and how this info is copied and passed on to further generations. Now, if you want to sum that up very, very quickly, I see a very key word, and it's repeated. So I'm going to circle it. Info. Info. Genetics is about information. We're going to find out, even though we've touched on it before, that DNA is the recipe to build all of the proteins that your cells need. So in studying genetics, we study how this DNA is carried in a cell, how it's copied, and how we pass it on to further generations. Now, we can talk about a genome. You may have heard of, the Human Genome Project. The genome is all, and that's very simply it. All the info. So if you went to a library, right, and you saw all these books on the shelves, every page, the appendices, the forewords, the main text, all the chapters, the headings, everything, every bit of the words in the library would be the genome, all of it. However, to study a genome is sometimes not only daunting but impossible, and you need to break it apart, and so does cells actually break apart this information. And it's broken apart into physical structures. And you say, well, wait a minute, that sounds very similar to hey, that sounds like a book. Yeah, that is a physical structure which carries information. It's called a chromosome in a genetic sense. So think about the genome being all the books in the library, and each one of the books would be a chromosome. Now again, the chromosome is nothing, the book is nothing unless we can get inside and read the information, right? So the chromosome is just the physical structure which carries the information. If you want to read the book, we got to open it up, right? So here's our book that's open, and now we can get to the next term, a gene, a sequence of DNA, and sometimes you'll see it written, GACCTAGC, there, that's a sequence of DNA that determines the order of amino acids. And remember, when we're trying to build a protein or a polypeptide, which is what we're going to do here, we need a chain of amino acids linked by peptide bonds. If you want to use our book analogy, here's our chromosome down here, and then inside this book would be, GATCCGT. I'm just making up letters right now, GAAAGTC. There's a gene. And each page would have another gene, and more genes. You can think of the genes as recipes, and I'll write that right here. Recipes to cook up proteins. So we could think of this as a normal library, or we could think of this as a cookbook library, and that might help us when we do some further analogies. I'll be using some kitchen terms because we're all familiar with cooking up some food. All right, so genetics, info, pass the info. Genome, all the info. Chromosomes, the physical structures which carry the info, a book. And then the genes is the actual recipes inside of the books that we will read at some point to create our thing we're cooking up, a protein in this case. In modern cells, information is transferred this way. So we've got 3 big terms here: replication, transcription, and translation. These will be the next 3 talks that we will do. This one I'm going to deal with replication. The next talk will be transcription, and then the third one will be on translation. So we know DNA is the info, and we know it's stored in the nucleus of our cells, and we know it's stored as genes. We just saw that with our library analogy. Remember the cell cycle. If we want to use this DNA, like during G1, gap 1, gap 2, if we want to use this info during interphase, we can't just go straight from DNA to protein. That would be great, but that's not how it happens. If we want to do this as cells do this, we have to go to a middleman called RNA before we go to a protein. Let's recall DNA structure. Let's do it quite briefly. Remember, there is a sugar phosphate backbone. And so I'm just going to put some sugars and phosphates here to remind us of this. And if you want, you can put the sugars in pentagons to remind yourself that it's a pentose sugar. And we can remind ourselves the D here in DNA tells us the sugar is deoxyribose. But the point is, I have a sugar phosphate backbone. And then hanging off the sugars, right? Hanging off the sugars is some sort of base. It could be, a G, an A, a T, or a C if we're talking with DNA. And just for fun, I'll put a G here because he looks lonely sticking out like that. That's great. So we have our sugar phosphate backbone. We have the bases sticking up. And then, of course, you'd have another one. You'd have a double-stranded molecule because DNA is double-stranded. Same thing, sugar phosphate, sugar phosphate, sugar phosphate. And just for fun, I'll circle all the P's just so we can pretend we're doing it for real. Hanging off the sugars again are the bases. And if you can remember base pairing that we talked about, G always went with C, A goes with T, T goes with A, C goes with G, and G goes with C. If you need a little booster on this, go back and check out my talk on nucleic acids, and I go more slowly through this detail. But that's a reminder for us, if we've watched the series, what DNA looks like. Yes, we will, of course, triple bond with hydrogen bonds, the C's and G's. We will double H bond the A's and T's based on the rules we learned a long time ago. So nothing has changed. We're just recalling this information so we can picture it in our mind. Now there is going to be one new thing though. We're going to find out that these strands don't actually run the same direction. Whenever we draw the pictures of them, they look like they're running the same direction, but they're actually going to be opposites. And these are going to have things like, you're going to see a notation like this. 5' is how you would read that. And then this would read 3'. And if you followed this 5 over to the other end, you'd see that this is a 3'. And if you followed this over to the other end, you'd find this is a 5' end. So we haven't mentioned that part yet. I've been keeping it from us and saving it for when it's necessary, and it's going to be necessary now. So let's look at this backbone structure a little more, this 3', 5' business. You have two roads, one going this way and another going this way too. We would call those what? Parallel. Right? We would call those parallel. If the backbones ran the same direction, we'd say there was parallel backbones in DNA, but they don't do that, right? We said one of the strands runs this way and the other one runs that way. So they're anti-parallel because it shows the DNA, the second strand, completely, totally, physically upside down on top of the other one. And it's a really good way to think about this. It's not just in reverse, it's inverted. 5' end. I'm going to get rid of this one bubble because it's kind of getting in my way. 5', remember we saw that a minute ago? 5', here's how do you remember it. Phosphate 5, phosphate 5, phosphate 5. The phosphate group is at the 5' end. And if you go over to the other end of the backbone, you see this 3' end, and on the 3' end is this OH sticking out that's part of the sugar. So 5' is phosphate, 3' is sugar. However, if you head over this direction, notice there's not another 3. These are not parallel structures. These are anti-parallel. So you should find, if you find a sugar on this end, 3', you should find a 5' phosphate on this end. And then follow it down here, finish the rectangle. You should find again at the 3' end an OH group, a hydroxyl group hanging off of the sugar. So the DNA strands run anti-parallel. Notice how one backbone is upside down relative to the other. A hydroxyl group, OH, part of the sugar is the important part, is sticking out of the 3' end. No matter which direction you're on, the 3' end has the sugar. That would mean phosphate 5, phosphate 5, phosphate 5. The phosphate group is at the 5' end. Why is this important? Well, we're going to find out very soon that enzymes are not very smart. In fact, they're not smart at all. They have one job, and that is to move forward. And they only move forward in one direction. The point is the enzymes can't go backwards. So one of these strands, when we start to copy this DNA, will be very easy to copy. And the other strand's going to be what we call a pain in the butt, right? It's going to be a multi-step mess to fix. Okay, so let's get into the details of replication. We say that replication is the exact or faithful copying of each strand. And remember, we got to copy each strand, so all of the DNA will be copied here. Quite simply, when you're doing replication, every bit of the DNA, even if it's billions and billions of letters, all of it will be copied. Remember that it happens during the R or S phase, during interphase, before mitosis occurs. Makes sense. If you're going to do mitosis and give a set of info to your offspring, you want an entire set. So we're going to copy the entire DNA molecule during R phase. That's why I like calling it R phase, because it reminds me of replication. Just to remind you, the S means synthesis. We're going to synthesize DNA. Same thing, but just a different term for it. What's the point? Identical copy. Remember, we're having an offspring cell here at some point. We're going to do mitosis. We're going to split the cell. We want our offspring to have a full set of info that's exactly like we have. So they are our genetic clone. Because base pairing is specific, and we learned, A with T and G with C, each strand can serve as the template. Template is just something you're going to copy for the opposite strand, even when separated. And that might not make sense when I say it right now, but let me put some details here to kind of help you figure this out, and then we'll run through the actual mechanism. It used to be thought at one point that replication was a conservative event. And if you want to understand that, just check out the dude over here with the copy machine. Now let me show you this. If you were to take a piece of paper and you put it in the copy machine, what do you get out of it when you're done? Well, you get your paper back, don't you? You get your, what we call the original. I'll just put that's the original, right? You get your original back. But also you get a copy. And I'm just going to draw the copy in a different color so you tell. You get a new copy. That's the way a copy machine works. That's a conservative event, meaning you're conserving the original information. You get the original back, perfectly intact, and you get a new copy back. Looks exactly the same. It's not exactly the same because it's not the same piece of paper, but the info on it is the same. Now remember, this is not how it works. This is not how DNA replication. It was once thought that's how it worked, but we now know it does not. So how does it work then? Well, it's a semi-conservative event. So to show you this, let's take some actual DNA, just like this. There's some DNA. We got our two backbones. We have our base pairs in the middle there. There's our ladder picture we saw way back when we talked about DNA. And imagine this. Imagine that we're going to, it says, even when the strands are separated. Let's separate the strands. Let's cut those. Let's cut those hydrogen bonds. I'll tell you who does that in a little bit, but that's all I've done. I've taken this original. Remember, we're calling it, let's call it old news, right? It'd be easy to call it old. And we've got some of that old news there and some of that old news there. We've just ripped it in half. And now we're going to copy each strand independently of the other. So we're going to lay down some new DNA here and here and here and here and here and here and here. And then we'll also lay down some new DNA on top of this old strand. So what we get when we're done is a DNA that looks exactly like the other DNA. All the bases match. We're going to run through the base pairing to show it to you, to prove it to you. But the results are two DNAs that are identical, but each one is half old and half new. This is the concept of semi-conservative replication. That's the way that it goes. It says it right there. Each new molecule contains one old strand and one new strand. And these are the terms I'm going to use to deal with these strands. All right, a little animation of this happening. Remember, this is our DNA originally, and we want to replicate it. How much are we going to replicate? The entire thing. Are we going to conserve this structure? No, we're not. The first thing we're going to do is split and then base pair. So let's do it. Split, and then let's base pair. A with T. These are new stuff, right? If this is the old stuff, then this is the new stuff we're laying down. A with T, G with C, and then let's do it again. This is old stuff. This is new stuff we're laying down. We're just base pairing G with C, A with T, T with A, G with C, and we got it, right? At the end of the day, notice how this strand here looks exactly like that strand. Just read it going up, T, G, C, A, T, G, right? It goes the same way. And this guy looks exactly like that guy. So we do have two DNAs. They are identical. I mean, notice the pairs there, GC, GC, TA, TA, AT, AT. They're all matched. They're all the same, but each molecule now is half old and half new. That's the concept of semi-conservative replication. All right, so what we're going to find out is that replication enzymes are gigantic. And if we want to get them into the DNA so they can actually get to the bases and work on the bases, we're going to have to open up the DNA so these big giant enzymes can get in there. In doing so, we're going to create what's called a replication fork. So it reminds us here, before the very large replication enzymes begin copying anything, DNA needs to be unwound by an enzyme called topoisomerase. Usually topo is enough. There's not any enzyme out there I know that's on standardized tests that starts with topo except for topoisomerase. And then unzipped by a helicase. So therefore, replication will occur at what's called a fork. Let's draw that out real quick. Let's draw some DNA. I'll show it all kind of coiled up. Show it being opened up. I can do that again, no problem. All right, now you can kind of see what I'm talking about. So let's put some bases in here so we can make it look like some real DNA. And there's a separated strand. And there's a separated strand. Kind of looks like an alligator mouth with all the teeth sticking out. And oftentimes in classes I would refer to this as the alligator mouth. This is the replication fork. Now we're not doing any replicating yet. Remember, this is just preparation, preparing the fork. So right here, and I'll change to a different color so we can see them a little better. Right here, looks like we have an enzyme, and we do, it is an enzyme. He's moving that way, and he's unwinding the DNA as he goes. This is topoisomerase. He's unwinding the DNA. There will be behind it another enzyme in here called helicase. And I'll just put an H here. This guy up here, helicase. He's unzipping. What does that mean? He's separating the bases from each other. He's ungluing them. He's cutting the hydrogen bonds that are holding them together. Sometimes you may find, even though I don't make a big deal about these, you may find some little proteins glued to these single strands of DNA. Remember, the DNA is now single-stranded since we separated it. Two separate strands, and they want to seal up. They want to fall apart. They don't really like that. So these are called SSBPs, or single-stranded binding protein. Oops, I forgot my R. Single-stranded binding proteins. These will align themselves near the fork, but on the links of the two strands to stop them from resealing with each other, to stop them from falling apart or snapping off or something like that. Basically to stabilize the complex there. All right, once we've got the fork open, we can now start doing the copying. This is going to require the work of many enzymes doing many different jobs. So I made the point that enzymes are great at what they do, but they're kind of stupid. They don't know how to go backwards, right? If I could reinvent the universe, I would just simply make enzymes that would go backwards, but that's not the way that it is. So synthesis occurs in a specific direction. There's an enzyme that does this, again, doesn't know how to go backwards. Very good at doing what it does. It says it lays down deoxyribonucleotides, which is great. And if you want to make DNA, you need to lay down deoxyribonucleotides. That's telling us we've got deoxyribose for the sugar in the backbone. And we're going to have T's in here, GATC, instead of any U's that would find in RNA. So the enzyme is DNA poly. What does DNA poly do? Sometimes I just call it poly. And sometimes you hear me call it she or her because it sounds like I got a girl's name, Polly, right? So DNA poly, DNA polymerase, is the enzyme that's responsible for laying down the nucleotides. Problem: poly only works one way. And you have to keep this correct. It's easy to flip it, but it always, only, forever works 5' to 3' along the new, or what we call growing strands. Not along the old strand, along the new strand. And remember, they can't go backwards. So one of these strands is going to be easy to copy. This is going to be called a leading strand. And it's very simple. DNA poly will sit down at the front of it and continuously synthesize toward the fork. There's the key. If you can find the strand that's being synthesized toward the fork, 5' to 3', you're done. You drop down poly, you tell her to go, poly copy, 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 all the way to the other end, even if there's millions and millions and millions of bases that need to be copied. Okay, so what? You say, well, it's the other one that's the problem. It's this lagging strand. So this is the issue. The enzyme doesn't go backwards here, and we haven't opened enough of the DNA to start on the other end. So what we're going to have to do is synthesize, first of all, away from the fork, discontinuously, meaning in pieces. We'll do a small piece away, and then another small piece away, and then another small piece away. And then we're going to have to stitch up all those holes we've left in our replication. And that's what it says at the end there. It will require extra steps to replicate. All right, lagging strand. Again, this is the one that's a problem. This requires a primer. And the primer is going to be made of RNA. That's strange. We're trying to make DNA. Why are we putting RNA here? Well, we're going to use a primase, which is an RNA polymerase. It's going to lay down ribonucleotides, not deoxyribonucleotides. So this looks to be a mistake. This is just the way that the system works. It's a little weird, but it seems to be a mistake. And it is, kind of. We don't want RNA primers in our DNA. We want DNA to be copied to DNA. So we're going to have to fix this problem, this lagging strand problem. We're going to have to fix it as soon as we can. How are we going to fix this? Well, Polly will finally figure it out, like, oh, whoops, we've got RNA there. I need to fix them. So she's going to find and replace these short primers and replace them with DNA fragments called Okazaki fragments. But they're not going to be all together. They're going to be short and discontinuous. So therefore, we're going to need to close the gaps. The gaps are closed later by a ligase, another type of DNA polymerase. Before we start, right, we have to do a little bit of a thought experiment. And all my pictures are going to keep this same system, but we have to know, before we begin, who's leading and who's lagging. Let's do a thought experiment only right now. Let's take some DNA. You know, we can do this game if you want. We can make a squiggly, then open it more. Okay, I'm just making my alligator mouth again. It doesn't have to be pretty because we're just doing a thought experiment. And I'm going to go ahead and label this 3', and I'm going to label this, of course, 5' because I can't have the same thing. They have to be opposites. Now you could talk about right here, you could have topo. Remember our friend topoisomerase, he's unwinding. You could talk about our friend helicase, unzipping. You could talk about single-stranded binding proteins along the length of it to stabilize this complex. But we're more interested now in thinking about which strand is the leading strand and which strand is the lagging strand. The only rule we've known so far is, well, enzymes only work one way, 5' to 3' of the new strand, and leading goes toward the fork, lagging goes away. So let's think real quick. If we were going to build some DNA on top of this DNA, it would look like this, right? Right? And if we were going to build DNA on the bottom strand, it would look like this. Just like that. We just added some DNA. So let's complete our picture. If this says 3', then this here is 5'. And if this down here is 5, then this end is 3. And if we go back up here to this 5, that means this is a 3 and this is a 5. And our rule was enzyme polymerase only always works 5' to 3', 5' to 3' of the new or growing strand. The blue guy here in this case is the new strand. The yellow stuff's the old stuff. So I see that this enzyme going 5 to 3 here is going away from the fork. This must be the lagging strand. And this one here heading toward the fork, this must be the leading strand. And we haven't copied anything yet, right? We haven't done any copying whatsoever. All we've done is set up our system. We know now which way is leading and which way is lagging. And to be fair, on most images, on most books that I've ever seen, leading is on top, but don't get that in your heads. You can flip it easily. You could have an image where you see the fork is built like this, and then there is no up or down. It's either right or left, so be careful on that. All right, now we're going to use the same information on the next pictures. Leading going to be on top, lagging going to be on bottom, and I will have the prime markers there. But let's actually run through the process. Let's actually do some replication now so we can see what happens. So again, the leading strand in our case was the top one. I've actually put some G's, A's, T's, and C's here so we can see what to do. And let's keep this super simple. If the top is leading, this guy up here, I know what I'm going to do. Remember, the fork's over here somewhere. I can't see it in this picture, but I know the fork is here. And I know the top strand, all I need to do to get this to copy is to plop an enzyme here, and this is going to be DNA polymerase, and it will run toward the fork 5' to 3' of the new strand. Hold on, I got to deal with the dog. What do you want? Stop it. Come in. Sit down. Stop scratching. Okay, 3, 2, 1. Now, what bases will be here? Well, again, you can go through and do this on your own if you want. But if it starts at this end, T goes with A, then A goes with T, then C goes with G, and T goes with A, and G goes with C, then a G, then a T, then an A, and you can keep on going and keep on going. That's the point. DNA polymerase will lay down DNA nucleotides, deoxyribonucleotides. She starts at one end and goes all the way to the other end. All the way. As long as the fork is opening up more and more and more, she's going to follow topo and helicase and just go all the way to the other end. And we're done. That's it. It takes one enzyme, plop it down at the beginning, and push it forward all the way. That's easy, like I said, no problem. But what about this dude down here? That's the dude that's going to be an issue. Because poly goes this way, we're going to have to do this in steps. We're going to have to do a little synthesis, and then a little more synthesis, and then a little more synthesis, and then we're going to have to fill these gaps in somehow. And that's going to be the story we tell on the next slide. All right, so let's check it out. Let's put it like this. Step number 1 involves using a primase. You see, that's the name of the enzyme, primase. It will lay down as it says in the text a few slides back, you can check it if you want to, it will lay down primers. Now remember, the problem here is that those primers are RNA. So let's show that happening here. Let's put one right here. Tick, 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 tick. There is a little primer. Now what will it read if I'm going to read the bases? Well, C goes with G, A goes with T, but wait a minute. This is RNA. So it's going to be a U that's put there. And same thing with this A. There's going to be a U put there. And then a G, and then an A. Okay. And that right here is called a primer. That little red thing that I've drawn, a little short piece of RNA. Sometimes it's said on tests, a short, discontinuous sequence of RNA. It was laid down by primase. You could go further down here and do it again. Let's say right here, we're going to drop another primer. Right there. There's our primer. What will it read? Well, this will be a C, a U, an A, a G, and a C, right? We're just doing base pairing. Nothing fancy. There's another primer laid down by a primase. And then we can scoot up more. Remember, we're laying these down this direction, though. We're laying them down away from the fork. Same thing here. Let's put another primer here. A goes with U, C with G, A with U, A with U, and C with G. Okay, another primer. Again, we're laying it down away from the fork. Just to remind us, the fork is this way. This is the first stage called priming. Now remember, we have to fix this problem. We don't want RNA in our DNA. So we're going to have to solve this problem with another step. How about step 2? Okay, let's take some yellow and talk about step 2. Step 2 I call find, replace, and extend. It's not exactly the way that books deal with it when you read the textbook, but I find that this way is easy to nail the questions that are asked on most standardized tests. So step 1 is a priming step. We're going to lay down primers with a primase. Problem, it's RNA. Step 2, we need to find that RNA. We need to find those primers and replace it with some DNA. And then maybe extend the chain just a little bit. Right? Maybe extend it just to give it a little bit, get it a little bit closer to the next primer so we don't have such big gaps in between. So who are we going to have do this? Well, we're going to hire an enzyme that should have done this in the first place. DNA polymerase. DNA poly will find, what is she going to find? Well, she's going to find a primer. So I've shown a poly, a DNA poly, not an RNA poly now, a DNA poly sitting on the primer. And what she's going to do after she found it is replace. Replace what? Replace that backbone. First of all, replace the backbone with DNA, right? With deoxyribose. We don't want ribose in our backbone. So she's going to clean the ribose out and put deoxyribose. And then she's going to turn these bases into actual DNA bases. So this U will get turned to a T. And this U will get turned to a T. This will stay a G. This will stay an A. And then poly will add a few more bases just for fun. C, T, A. Now it's a DNA, right? Poly, so it's going to add T's. It's going to add the right thing. So there's a find, replace, extend. Let's do it again. I see another primer up here. What's going to happen? DNA poly will find it. Remember, and poly's moving this way. Find, success. Replace. Replace what? Replace the backbone. Replace the ribose with deoxyribose. And then replace the bases that are RNA. So the C stays, but this goes to a T, to a T. The A stays, the G stays, and the C stays. But let's add some more. Let's add a T, a G, T, C, right? Just base pairing. So there's another example of a DNA poly finding a primer, replacing the primer with DNA, and then extending the sequence a little bit longer. One more time just for fun, right? Here's a third primer. What's going to happen? DNA poly finds the primer, sits down on top of it. Replace. Replace what? Replace the backbone. Replace ribose with deoxyribose, but also replace any bases that are still RNA bases. So the U goes to a T, the G stays the same, the U goes to a T, the U goes to a T, and the G stays the same. There's find replace. Now let's extend. Come out here a little bit. That's a T, that's a C, that's a C, and that's a G. Let me erase some of this so we can see step 2 as it's completed. Wonderful. But we still have huge gaps in between these sequences. And that's why we need the third event to clean up those gaps. We need a third step that's going to involve another enzyme. So first of all, remember we had our primers laid down. And those primers were little short, discontinuous sequences of RNA. But now what do I have down here? What do I have here? I have a short, discontinuous sequence of DNA. Aha, it's a little longer than the primer, but it's still short. And it's still discontinuous. Now this is called an Okazaki, named after the person who discovered it, fragment. An Okazaki fragment. There's another one here. Here's another one here. So after step 2, we're left with Okazaki fragments, and we need to stitch them together. So this third step, you can call it, stitch. Let's stitch up the gaps in between the Okazaki fragments. And in order to do this, we need a third enzyme. Usually this enzyme is drawn like an arrow. On a lot of books copy this, so I'm going to do it too. It's drawn like an arrow in the gaps between the Okazaki fragments. And this guy is a ligase. I know it's an enzyme, ligament, ligature, something that's being tied together, right? So a ligase is an enzyme that ties Okazaki fragments together. And it is another type of DNA poly. Again, you can get in DNA poly 1, 2, 3, and so forth. I'm going to skip all that, and I'm just say this is a form of DNA polymerase called a ligase. And the ligase, again, working away from the fork, will add all these extra bases in between. I don't want to copy them in there. You can practice if you want. You know, this would be an A, this would be a T, this would be an A, and so on. We're going to add DNA. And I'll do it here too. I'm going to stitch together those Okazaki fragments that were left after step 2. Now we're done. Now we're done copying that lagging strand. Oh boy, we made it. A little painful to do the lagging strand, but I think we got it. If you enjoyed this video, watch some others. Certainly thanks for watching that one. I'll see you on the next one. Okay, bye-bye.